right, welcome back. So last time we talked a little bit about Koch's postulates, kind of identifying that microbes could be the causes of deadly diseases, which was actually thought to be kind of weird, right? There's all these little microscopic things. How can they harm us, these giant organisms? But through the work of Koch and his postulates, we have a set methodology of identifying a bacteria, a specific bacteria or microbe that could cause a specific disease. So in this review question, you are attempting to show that bacterium X, right, just, whatever it is, we've identified something new, causes a respiratory disease in your laboratory mice. Why would you want to do this? Well, maybe we have a new respiratory disease going around. We all have experienced that, unfortunately, with COVID, and we need to identify, is this bacteria actually the cause of it? In the case of COVID, that was a virus, which we'll talk about more in this course, but that's different from a bacteria. In this case, we've isolated the bacteria from sick mice and grown that into a pure culture. So remember, a culture is just like a growth of microbes. We'll do this in the lab. You can grow them in broth. You can grow them on plates. We'll do both. But this case, we have a pure culture, which means we only have that single species of bacteria in there. So what would be our next step to fulfill Koch's postulates? Would it be A, determine the shape of the bacteria using a microscope, B, sequence the bacterial genome, C, inoculate healthy laboratory mice with the pure culture, D, vaccinate healthy laboratory mice against the bacterium, or E, isolate the same bacteria from healthy laboratory mice? Go ahead and pause the video. Think about that one. All right, in this case, the answer would be C, inoculate healthy laboratory mice with a pure culture. So remember, Koch's postulates, we take sick individuals, in this case mice, and we isolate the bacteria from them, and we grow it in a pure culture. We think that bacteria is causing the disease. So what do we need to do next? We need to take that bacteria, introduce it into healthy mice, and see if they become sick. That helps establish that chain of infection there. Determining the shape would be interesting, but doesn't really tell us if that's causing the disease. Sequencing the genome, well, when Koch was doing this, that wasn't possible. He didn't even know about DNA. It is possible, but still, again, it doesn't tell us if it's causing the disease. C is our answer. Vaccinating, we can't make a vaccine if we don't know that this bacteria is causing the disease. And isolate the same bacteria from healthy laboratory mice, well, that would rule out bacterium X being the cause, right? If we got the same bacteria from healthy individuals, it's very unlikely that that's causing the disease unless something crazy is happening. So let's dive into it. Okay, so we're going to finish up chapter 1.4 and 1.5. They're very quick, just overviews. So in this course, a lot of us are really interested in medical microbes, but microbes are everywhere, and particularly in our environment, they play critical roles in the natural ecosystems and also in our own evolution here, as we'll see. So we can grow about 0.1% of microbe species in the lab. That's not a lot, right? That makes things really hard to study. Microbes, by definition, are microscopic. So if you can't grow them, they're really hard to find and kind of watch what they're doing and things like that. So we need to grow them in the lab to study them and figure out what they like and what they don't like, what they need to eat, things like this. But we can only do that with about 0.1% of the species that we believe are out there. In some cases, there are microbes that grow off what we call non-organic nutrients. This isn't organic like organic farming. This is the chemistry organic, which means carbon-containing. So we are carbon-containing organisms. We have all kinds of organic molecules in us. And life as we know it, or as we generally know it, generally takes organic material. You eat a plant, which has organic material in it, and you get those nutrients from that plant. And the plant, it gets roots in the soil might take up organic nutrients from things that have decomposed, that fungi have decomposed. But there are some organisms, like things that can live at the bottom of the ocean, that can live entirely off non-organic things like hydrogen sulfide and these other strange chemicals. So there are all kinds of crazy ways that microbes can live outside of what we normally think of. And to study microbes, one of the first people to approach this systematically was Sergei Winogradsky. He developed what we call an enrichment culture technique. So enrichment cultures, you put specific things into what we call the growth medium. These mediums, that could be like an agar plate or something. You might put certain nutrients in there that a bacteria needs and exclude other things that they don't, so that you can grow specific types of microbes. Now, why do we care about this? Well, it's interesting, right, to go out and grow things from the environment, see what's out there. But medically, this is super important because your body is covered, and actually throughout it, there's tons of microbes, right? You got all kinds of microbes in your gut. And when you get sick from a microbial disease, it's usually one specific type of microbe. You want to identify that because we need to give you the right drugs to treat that, right? The right antibiotics. And so if we take something like a stool sample, that's poo, right? And we put it on a petri dish, there's a lot of bacteria in there, a lot of different bacteria. Most of them should be there because we have our own microbiome, right? You got all these things in your gut that help you digest your food and do all kinds of important tasks. But somewhere hidden in there is some sort of microbe that is causing sickness. And we'd like to be able to separate out the good microbes from the bad microbes. We can kind of do that using what we call enrichment cultures. So we might be able to grow a certain group of bacteria called the gram-negative bacteria. That includes E. coli. And we want to exclude a whole other group of bacteria called the gram-positives, which normally don't cause disease in our gut. So we'd grow our bacteria on a plate that has specific nutrients that only allow the gram-negatives to grow, but exclude those gram-positives. They can't grow on it because it doesn't have the right nutrients for them. We're actually going to come back to this concept later in the course, and we'll even kind of look at this in the lab. This is a common thing that medical microbiologists do. All organisms that we've studied have a microbiome. This is all of the microbes that live on and in an organism. And we believe that most of these are really important to healthy living. We can grow mice in a completely sterile environment where they don't have any gut bacteria. We birth them in a sterile environment, and they have digestive problems. If you think about things like cows and termites that eat high cellulose content food, right? Cows eat grass. If you eat grass, you don't get much nutrient value from it. But cows have bacteria in their guts that can break down the cellulose in plants and give the cows nutrients. What do the bacteria get in return? They get a place to live. They get a little bit of nutrients out of it as well. A nice home there. Same thing with termites. Termites dig through wood and they actually eat the wood. They can't digest it, but they have bacteria in their small, tiny little guts that break down the cellulose in there and give them nutrients from it. You too have gut bacteria that helps you. And this is a crazy thing to think about, but sometimes when people have really bad infections, we give them very strong antibiotics, which has the effect of actually clearing out their gut microbiota. And when that gets screwed up, people can have digestive problems. So we're curing their disease, right? Say you have like an infected finger that's, gonna fall off basically. You want to treat that, but we have some side effects of that. And there's actually a procedure that the FDA has approved called a fecal transplant, which is just what it sounds like. It is taking poo from a healthy individual and introducing that into their gut. There are two ways to do that. You can go through the mouth or through the other way to get to the gut. But that is an actual procedure that can be done to help bring those healthy microbes back into our guts. Now, I said microbes are important to you in terms of how you've evolved into a eukaryotic organism. We'll talk more about what a eukaryote is in a bit, but we are eukaryotes, right? Plants, animals, fungi, these things called protists. We're eukaryotes. If you look at a cell here, right, here's a plant cell. They have a cell wall. We don't have it. We have a cell membrane. But here we have all these little things inside our cells. We have a nucleus that contains our genetic material. We have these little things called mitochondria that you've probably heard of. Those are the powerhouse of the cell, right? They create a lot of energy for us. And in plants, they have this special organelle called chloroplast. That is where photosynthesis occurs. That's where the green pigment is that makes plants green. Well, the funny thing is that Lynn Margulis proposed that these, what we call organelles, the mitochondria and the chloroplasts, they looked a lot like bacteria when we look at them under a microscope. And she proposed that we had this process called endosymbiosis. So what does that mean? Endo means inside and symbiosis means living together. So inside living together. Well, so what do we think happened? Well, we think very early on in evolution, we had just prokaryotes, bacteria, things that don't have any organelles, small microbes. And at some point, we had this pre-eukaryotic cell that absorbed some sort of bacteria into it, and they started living together. And eventually, they basically merged. They cannot live without one another anymore. The mitochondria, as it's become to be known, that provides energy for the cell. It does a lot of chemical reactions for our cells, and in turn, the cell provides it with food and a safe place to live. We think the same thing happened with chloroplasts. Cyanobacteria, photosynthetic bacteria, got absorbed by a cell through this process of endosymbiosis, and they began living together, and that's where we got our plant cells. We'll come back to this a little bit later, but this is really, really interesting, right? We have these prokaryotes out there, and we've actually absorbed them into our own cells to do things. So part of you is bacteria, basically. Okay, that's it for section 1.4. We're just going to dive right into 1.5 because it's real quick, talking about one of my favorite subjects, which is DNA. So this is what really got me into science in high school. I thought DNA was just the coolest thing ever. Doing PCR, which is something that we'll get to do in there to replicate DNA in a test tube. It's just the coolest thing. But we haven't actually known about DNA for that long. Right now, we're looking at about 70 years of really understanding this. And when we think about scientific knowledge, that's not really that long. And we're still learning things every single day. So we're going to talk about how the structure of DNA was discovered briefly, how DNA information is used and how it can help us in terms of medicine and industry. And then we'll briefly touch on kind of using microbial biology to eventually let us maybe edit human DNA for therapies to help things like genetic diseases. So the discovery of the structure of DNA, it's a long, convoluted story. There were a lot of players. If you're really interested in it, I love to talk about it. There's lots of information out there. But long story short, a lot of people were looking at this and had a lot of ideas. One of the people who was doing this was Rosalind Franklin, who was looking at DNA using a very special type of microscope called an X-ray diffraction microscope. And it produces basically a scatter pattern. You shoot X-rays at a crystal of DNA and it makes this X pattern. And I was very fortunate many years ago when I was looking at graduate schools to see one of these original pictures that she produced. I went to Oregon State University, and Linus Pauling, who you may have heard of, he's a very famous chemist, he donated all of his stuff when he died to Oregon State. So there's this huge room, bigger than our lab room, full of all the things from his life. And one of them was this image from Rosalind Franklin of this X-shaped pattern, which, if you're an X-ray diffractionist, means that DNA is in a helical shape. Now, there's some contention here because Franklin worked with a guy named Maurice Wilkins, and Wilkins shared this information with who we know as discovering the structure of DNA, Watson and Crick. So they took his information and some other people's information and other people's information and combined it all together into a model of DNA, which is our double helix. We have our 4 letters, right? A, T, G, and C on the inside in the rungs of the ladder, and then the rails of the ladder twist around. We'll learn all about DNA later in the course. But Franklin didn't probably get as much credit as she should have received, and a lot of people don't really like James Watson. He's not a very nice guy. Crick, by all accounts, was a very nice gentleman. I've heard from people who've met him that he was very kind. But this whole thing, right, they didn't do any experiments. Watson and Crick just used some math and other people's information to put this together. So some people were a little bit upset that they didn't get credit for their work. Unfortunately, when Watson and Crick and Wilkins got the Nobel Prize, Rosalind Franklin had died. So we don't know if she would have got the prize. You can't receive it after you're dead. So unfortunately, we'll never know. But we're starting to recognize her contribution more and more to this process. Now, knowing the structure is one thing, but being able to read DNA, that's like the big breakthrough. And it took us a long time to do this. We couldn't actually read DNA until the late 70s. And we didn't sequence our first genome until 1995 when we sequenced it of this microbe called Haemophilus influenza. It took us longer into the 2000s to get the human genome, and we're still figuring out what it means. This string of letters, right? What do the A, T's, G's, and C's mean? We know some of it, but we're learning new things every day. The person who developed this technique is called Fred Sanger, and he developed a way to basically sequence DNA, and that has led to huge revolutions, right? Because now we can read the code of life. What have we used that for? Well, medicine, right? We've used these discoveries with DNA and proteins and things that they code for to make vaccines. Polio is one of our gold standards, although we'll talk about this a little bit. There are some re-emerging cases, but polio has pretty much been eradicated through vaccination campaigns, which is the result of microbial research and knowing how to work with DNA and proteins. So we have things like that. Our COVID vaccine has worked very, very well. It cut the death rate quite significantly. And other vaccinations, smallpox vaccination, we've eradicated smallpox. Hopefully it never comes back. Although we are seeing polio, we're having some issues where under vaccination is leading to reemergence of that scary disease, which causes infantile paralysis. Basically babies becoming paralyzed and not being able to breathe and then dying from that. But what about genetic diseases? Well, we have tools from microbes, things like you've probably heard of CRISPR, which is a thing we've stolen from microbes that allows us to cut and paste bits of DNA. This could help us treat cancer. Cancer is a disease that we'll talk a little bit about, but it doesn't really fit into this course because cancer is not a disease that's caused by a microbe or some sort of outside agent. It's actually errors in your cell's DNA that lead to unrestricted replication. And if we could fix those errors, we could potentially eliminate cancer, which would save millions, billions of lives over the time. Genetic defects. Now, this gets into ethical questions: is what's a genetic defect, right? Who's going to decide who gets to have this done to them? Or, children can't decide. There's all kinds of questions around this. But these technologies, they're not good or bad, right? It's how we use them. So we have to think critically about them. But they hold the promise, right? Any technology can be used for good and for bad, so we have to think about that. All right, we'll talk more about DNA in great detail. I just want to give you a little bit of an overview here, but that's a huge thing, right? Studying microbes allowed us to study DNA because they're so simple. They have so little DNA when we compare them to ourselves. That's why we sequenced a microbe first. So to finish up 1.5 and this chapter, we have, the structure and function of DNA. In the 20th century, we had a series of experiments that revealed that genome sequencing has really shaped the 21st century. That's when we started to really sequence a lot of genomes. And these microbial discoveries, right, they don't just help us treat diseases, but we steal things from microbes to use for other things. Biotechnology, insulin. We use bacteria to produce human insulin using these DNA technologies so that we can help diabetics. Microbiology, right, has all kinds of careers in health fields, right? Every time a nurse takes a sample or a swab from someone, we send that to a lab. Who does that work? A microbiologist in the lab. So it's really critical to understanding what is causing disease. And there's tons of careers opportunities there in both treating disease and preventing disease. That's the great thing. All right, so that was a big overview. I'm going to leave you with one of the things that I love about microbiology, which is microbe art. These streaks on here to make this nice painting of a sunset scene here, these are all bacteria that have been engineered with glowing proteins. So here's green fluorescent protein and blue fluorescent protein and red, right? So scientists, sometimes they like to have a little bit of fun, and there's an annual competition for this microbe art to create the coolest pictures using microbes. I particularly like this one. All right, that's chapter 1. On to chapter 2.